In this video, we're going to talk about various different mechanisms by which eukaryotic cells control regulation of their genes. So what does that mean? These are considerations as to when a gene is turned off or on, when the RNA is made as a result of transcription, or when the protein is made as a result of translation, how much of these molecules would be produced, and also the question of, okay, you've made these RNA and proteins, how long are they going to last? All of these things are regulated at various different levels. Now, when you compare gene expression between bacterial cells and eukaryotic cells, so prokaryotic cells, the process of translation and transcription are all continuous. So there is no separate region in the cell where transcription is taking place. So as soon as messenger RNA comes out as a result of transcription, ribosomes jump on and start making protein. So it's pretty simple, but that doesn't mean that transcription and translation are not regulated in prokaryotic cells. They do. However, in eukaryotic cells, because there is so much division of steps in gene expression, there are more and more opportunities for regulation of gene expression. And actually, that's one of the factors that contributes to eukaryotic cells and organisms being more complex. So here it is. During transcription, first an RNA is being made, which is pre-mRNA. This is the primary transcript. Then that primary transcript needs to be processed into mature RNA. This is a process during which you can regulate gene expression. You can also regulate gene expression during transcription itself. So you can decide whether or not this pre-messenger RNA is made or not. Then the cell can decide whether once the pre-messenger RNA exits the nucleus, it can decide whether or not it sticks around, how much of it sticks around, and whether or not it's being translated, how much of it is being translated, and when the protein is produced, there are decisions as to how much of it is made, how long it's going to be around, whether or not it's produced in an active or inactive state. So we're going to talk about all these mechanisms. So regulation of gene expression. This is a summary of how gene expression works in eukaryotic cells. So we have transcription here, formation of the primary transcript. That's the primary transcript that has exons and introns, and the exon and the introns need to be taken out. And then the RNA is also modified by the 5' G cap and a poly A tail, which finally gives us the messenger RNA. This RNA modifying step that removes intron is called splicing. And then that RNA is made to use for translation. So we're going to start with whether or not the decision whether or not the gene is actually accessible to even be transcribed, which brings us to the structure of our genetic material. So on this area, you see a chromosome. You've seen this before. So this is a highly condensed form of our genetic material. Usually, most of the time, our genetic material are in this untangled form where the DNA is wrapped around a complex of proteins, which we call histones. Now, depending on how condensed a piece of DNA is, it really determines whether or not genes are expressed, because usually genes are sitting on these intermediate steps in here, in between, which are not wrapped around the histone. So the question is, are these going to be accessible or not? And how does a cell regulate that? Well, in two ways. Either by DNA modification or modification of the histone proteins. With DNA modification, one of the ways that nature decides whether or not a gene is transcribed or not is by modifying a cytosine. The process is called DNA methylation. So basically, a methyl functional group is added to a cytosine. And when that happens, the region around this cytosine is going to be unavailable. The genes that are close to this methylated cytosines are going to be shut down. They're going to be silenced. Another way you can silence genes is by modifying the histone. Now, histones bind to DNA, and one of the things that it allows it to really nicely bind DNA is this N-terminal end that is very basic, and we know DNA is acidic. So that's a nice match between the different charges. Now, you can also decide how tightly these histones are going to grab onto DNA and kind of cling to them, deciding whether or not this surrounding region is going to be compact or loose. And you can do that by modifying this tail of the histone. So some of the ways that tails of the histones are being modified is by adding acetyl groups. Acetyl groups have a negative charge, so that negative charge is going to neutralize the positive charges on the histone tail, which you can imagine that when you neutralize these negative charges, then these histones won't be snuggling to DNA all that well. So therefore, you can imagine when that doesn't happen, the DNA opens up, becomes more accessible. Same thing with phosphorylation. Methylation, it tends to bring things together and shut things down. So now we're looking at two situations, genes switched on and genes switched off. On the top panel, you can see that the DNA is widely open, and the regions that are in here, genes that are in these regions can be expressed. And what makes that possible? Where the cytosines are not methylated and the histone is acetylated. These histones are acetylated. So the negative charge on the tail of histones is, if you like, neutralized. Now, if the cytosines become methylated, and if we remove the acetyl from the histone tails, then the tails are going to closely associate themselves with the DNA, and then it's going to make the DNA more compact. So therefore, the genes become turned off. So this is a very common mechanism that happens in nature. You may have learned or heard about epigenetic factors, properties that are inherited but has nothing to do with a change in the DNA. A lot of times epigenetic inheritance has to do with changing and modifying the DNA structure of cells, such that, for example, if a gene is turned off, then it's going to remain turned off in the next generation. So, as a review for yourself, if you want to test to see if you really understand this stuff, just think about one or more type of chromatin modification that can silence a gene, and what is one type of chromatin modification that can activate the gene. And the answer is obviously here. You can just test your knowledge. So another level of gene expression is by transcription activators. So let's imagine that this gene is sitting in an area of the chromatin that it's available. So let's say the chromosome is not condensed in this region. Now, this gene can be expressed at a basic lower level, or its transcription can be highly, highly enhanced. Its level of transcription is going to be highly enhanced, such that the cell is cranking up a lot of RNA out of this gene. So let's take a look at what's happening here. So this is the gene itself, the protein coding region. And when we discussed transcription, we talked about the promoter. This TATA box, the TATA, is kind of a signature sequence for promoters, to which a transcription factor binds and recruits the RNA polymerase. When we talked about transcription, in previous lectures, we learned that RNA polymerase, in order to find a gene to transcribe it, it needs the help of transcription factors, and this is what it's supposed to represent. So this will give you a basic level of transcription, not necessarily too high of a level. Now, there are elements on the chromosome, sometimes upstream of the gene, close to it, sometimes very far distance from the gene, which allows the cell to really crank up amount of RNA production from this gene. And these are called enhancers, and the way the enhancers work is through the function of other transcription factors. Or also you can call them transcription activators. So these transcription activators specifically recognize these DNA sequences that are in the enhancer region, and then the way that is thought that they work is they fold the DNA, and basically they recruit more RNA polymerase and this transcription factor that binds to the promoter and really concentrate these RNA polymerase and these factors into one region. So you basically drive formation of the RNA transcript to a very, very high level. So how do you regulate that? So first of all, you can increase the amount of activity, but you can also choose where you can activate this gene to higher levels. So depending on what cell you have, you can activate transcription of different genes. So what do I mean by that? So here it is, we have a liver cell and a nerve cell. And in all of the cells in our body, we carry exactly the same genes. But genes that are expressed in liver cells aren't really expressed in nerve cells, and vice versa. How does that happen? Well, the gene for alcohol dehydrogenase, which is involved in breaking down alcohol when we consume it, is only expressed in liver. But it is not expressed in nerve cells. And the gene for the neurotransmitters that send signals is active in neurons, but they're not active in liver cells. How does the cell decide that? Well, it really depends on the choice of the transcription activator that is expressed in the cell. So if we go back in here, if you can imagine that, let me bring my pen. So we have, let's say this is the gene for alcohol dehydrogenase. I'm going to write it here. And then you're going to have another gene down here, which has the TATA box, and then the enhancer sequences. Okay? So for the gene for alcohol dehydrogenase, this is the neurotransmitter, they would have different enhancers. So in this case, the enhancer is red, and I write it in a different color because it's a different sequence, different DNA sequence. And then in alcohol dehydrogenase, we have this enhancer that is a particular sequence. Now, each of these enhancers bind to a particular transcription factor. So the alcohol dehydrogenase gene, the enhancer upstream of the alcohol dehydrogenase gene and enhancing its expression, binds to these purple transcription factors. But let's say we make this blue, the gene for neurotransmitter, their enhancer, binds to blue transcription factors. Okay? So then, what happens? In a liver cell, only these ones are expressed. Only these transcription factors are expressed. Okay? But in a liver cell, these blue ones are not expressed. But in a neurotransmitter cell, let me change my pen color. In a neurotransmitter cell, it's a nerve cell. Okay, I'm just going to write N. In a nerve cell, these purple transcription factors are not expressed, but the blue ones are expressed. So you can see the cell becomes specialized in terms of the things that it can do by deciding which gene to activate. In liver cells, you activate the alcohol dehydrogenase because you have the right transcription factors to activate the gene. In nerve cells, you activate the neurotransmitter because you have the right transcription factors that activates the gene. So that's one example. We talked about post-transcriptional modification of RNA, and one important one is splicing, where introns are removed. So every gene has multiple introns and exons, and eukaryotic cells have the opportunity to make all sorts of different flavors of proteins depending on which exon they leave out. So you can start with one gene and make multiple different proteins. And sometimes you can even retain an intron that in other cases would not be retained. So that gives a lot of plasticity, a lot of flexibility to eukaryotic genes. And that's one of the factors that is involved in making us so complex. So what is a real-life example of that? Well, if we look at the muscle that makes our heart, there's basically structures in our muscle which are made of actin and myosin and this protein in here that allows the muscle cells in our heart to contract and expand. Now, this protein that is involved here in red is involved in creating that contraction. Now, this protein can either be formed in a short form or a long form. Now, in a normal individual, in a healthy individual, there is a gene that makes sure that our cells produces a shorter version of this protein, because there is an exon that it just removed. It doesn't make it into the final product. So you can see that these two are being excluded, spliced out. But in individuals who have a mutation in the gene that makes this protein, the splicing is skipped out. So therefore, the individual makes a longer protein. Now, in normal individuals, normal healthy individuals, the cell has enough tension in it. The cells have a tense enough muscle in them such that they can really nicely contract and expand the heart. But in patients that have this mutation in here, the tension is decreased. So therefore, it leads to a dilated cardiomyopathy. The heart muscles aren't as tense. You can think about this. If I give you an analogy, in this case, you can think about an elastic band that is pretty new. You bought it from the store and it's really tight, and you can contract it really well. But over time, that elastic band wears out, it gets soft, and that's analogy to this situation. So here's a real life example of alternative splicing, and when it's not done properly, it can have health ramifications. Now, when the DNA sequence of individuals were identified in eukaryotic cells, if you remember, we found exons and introns, and there's also all sorts of areas in our genome which scientists didn't really know their functions, so they called them junk DNA. So it was thought in the beginning, before we learned about their function, it was thought that these are just useless DNA molecules, and then you make this primary transcript that has these introns and taken out, and you might think of it that's, oh boy, what a waste of time and energy. But actually, it turns out that a lot of these introns actually play a regulatory role. A lot of the introns that are present in the genes of eukaryotic cells produce short RNAs, which are called micro-inhibitory RNAs. They produce microRNAs. And then when they're produced, the RNA folds on itself. It forms a hairpin, undergoes processing, and it produces something called a microRNA. Now, these microRNAs can do multiple different things. Sometimes they form a perfect match with their target RNA. Now, remember, this is coming from an intron, so it can target a messenger RNA, a primary transcript that includes this sequence. So it can bind to it perfectly, and if the binding is perfect, then this RNA is doomed to degradation. So that's one way the amount of a messenger RNA inside the cell is regulated using microRNAs. Now, in another condition, sometimes the intron doesn't perfectly match the messenger RNA. In that case, instead of leading this messenger RNA to being degraded, the translation is being inhibited. So this small inhibitory RNAs, their functions are involved in development, in cancer development, heart disease, kidney disease. These are just some few examples in which there is a role of these microRNAs in real life. Now, let's say the protein is being made. So the RNA is transcribed, the gene is transcribed, the RNA is being made, processed, and it's active, not degradation. You make a protein. Well, not all proteins are going to be active right when they are made. Sometimes proteins are produced in an inactive state, and then they needed to be modified, which is called post-translation. You need to activate the protein after it is made. And the cell can decide when this activation can happen. So a cell can make a protein in an inactive state and wait until it receives a signal, then it activates the protein. So an example here is for insulin. Insulin is first made as a pre-pro-insulin, which allows it to, so this signal sequence allows the protein to insulin to go into the secretory stage, then that signal sequence is removed, and then the pro-insulin is further modified during its secretion out of the cell into active insulin. So again, post-translational protein activation is one way that eukaryotic cells can control when a protein is made and when it's activated. We also have post-transcriptional control of RNA stability. So if you remember, when a messenger RNA is made, the cell adds 5' cap and poly A tail to them. But there's also sequences that are upstream of the gene, the sequences that determine the protein, and downstream of it, which are called untranslated regions. That's what UTR stands for. And these untranslated regions aren't just sitting here with no purpose. They actually bind to certain proteins, certain RNA binding proteins that can influence the activity of the RNA molecule. It can influence its stability. Another way of influencing the stability of RNAs and whether or not it can function, we mentioned, was by the miRNA molecules. We can also control whether or not a protein is made, whether or not an RNA is going to be translated or not. So an example is this protein EIF2, which binds to the small ribosomal subunit. Now this shows phosphorylation. This shows that this protein is phosphorylated. When EIF2 is phosphorylated, no translation happens. So you got the messenger RNA hanging around, but the small ribosomal subunit is tied up. EIF2 is stopping it from going and binding to the messenger RNA. But when EIF2 is not phosphorylated, then translation can happen. So a change in the phosphorylation state of proteins that bind to the ribosome can also regulate whether or not a protein is translated. It can also regulate its timing depending on when you're going to phosphorylate or dephosphorylate this EIF2 molecule. Finally, let's say the protein is made, but proteins, not all proteins have the same amount of half-life, and half-life is how long it takes before half of it is being destroyed. And that also can be regulated. And one of the ways that this is regulated by adding these short pieces of peptide to the end of proteins called ubiquitin. So when the cell decides it's time for a protein to go, it adds these little proteins with the power of ATP, adds these little peptides to the protein, and it tags it for destruction. So what happens to the proteins that have ubiquitin added to them? They are shuffled to a big protein complex that basically is a shredder for proteins. These ubiquitin tagged proteins are going to go through this complex and then just come out as amino acids. So that's at the end of the line for this protein.